Hi, my name is Manju Bhatt. I'm going to be talking to you today about calcium, which is an important mineral and a divalent cation in the body. Calcium plays a major role in a variety of body functions, including how our neurons communicate, how our muscles contract, and how our blood clots, among many other things. So they work with the organ systems to make sure that normal calcium levels are maintained in the body, particularly in the blood. Thank you.
Hi, I'm Tyler Allen, a PhD student at North Carolina State University in Comparative Biomedical Sciences. Today, I'm going to be talking to you about angiopolosis, an alternative mechanism of cell extravasation. Angiopolosis represents an alternative mechanism of cell extravasation. In this method, the cell will travel through the bloodstream and then attach to the blood vessel wall. The endothelial cells of the blood vessel will then actively wrap around the cell and remodel and push it from the inside of the lumen into the surrounding tissue, where it can exert its beneficial and regenerative effects. First, the stem cell will travel through the bloodstream, where it then becomes enlarged or attaches into the blood vessel wall. Once attached, the blood vessel wall will then extrude protrusions around the cell and then remove it from the lumen into the surrounding tissue. Once in the surrounding tissue, it can then exert its regenerative or therapeutic effects. Using intravital light sheet microscopy and 3D rendering, we're able to see exactly where the cell is in relation to the blood vessel and the vasculature, and the method that it uses to extravasate. Here, we have a time-lapse image of a stem cell undergoing angiopolosis. In this video, the stem cell is actively being pushed from the lumen of the blood vessel into the surrounding tissue, where it can then exert its beneficial or therapeutic effects. If you have further questions or would like to talk about this more, please contact either myself or my advisor, Dr. Kay Chang. Thank you.
So our next class topic is going to be on proteins and protein structure. The topic of this video is amino acids, and amino acids are the building blocks of proteins. And to give it a little bit more of a fancy terminology, proteins are polymers of amino acids. Let's see. So a peptide bond is formed through a condensation reaction. And it's called condensation, and I'm gonna have reaction as RXN. A condensation reaction is called condensation because water is formed through the reaction. So every condensation reaction shares that characteristic, that water is formed.
In today's pre-class exercise, we're going to look at the structure of a eukaryotic cell and its organelles. Organelles are the membrane-enclosed compartments that you find inside of eukaryotic cells. So by the end of today's pre-class exercise, I'd like you to be able to, 1, draw a eukaryotic cell and all of its organelles, as well as the features that we'll talk about today, and describe their function. And it's a dense region that's visible because it contains both RNA and protein. And this is where ribosomes are synthesized. Okay, now let's turn and look at some of the organelles and structures that are found in plant cells, but not animal cells. So here we have our generic plant cell. First thing I want you to notice is that you don't see centrioles in this plant cell, nor do you see lysosomes. Those are features that we only find in animal cells. The other things that you might find stored in a vacuole are chemicals, like caffeine, and also toxins that make plants toxic to some animals. And you also find pigments. stored in vacuoles. So this is what makes your flowers nice and colorful. Okay, so that wraps up our tour of the eukaryotic cell. In class, we'll look in more detail at the structure and the function of specialized cells.